Clarissa. Thank you.
Got it.
Great.
Okay.
Okay. So, oh, wait,
sorry.
Steve, let me introduce you.
Yeah, right.
We're not making you start without it. I'd like to welcome everyone to this morning's Ancestry and Diversity Forum. I'm really delighted to be able to introduce Dr. Steve Mack, professor at UCSF in the Department of Pediatrics, Division of Allergy, Immunology, and Bone Marrow Transplantation. I will admit, before joining ClinGen, I did not know Steve, even though I suspect that we've crossed paths in different ways. And Clarissa did a wonderful job sending out his resume. And so for those of you who didn't read it, Steve is really here today to talk about applications of HLA diversity for understanding human population relationships and how ancestry impacts the ability to identify donors for bone marrow and solid organ transplantation. And in particular, Steve is the co-lead of the RAD working group for the clinical domain, as well as co-leading the HLA working group for ClinGen. So we are delighted to have him come speak with us this morning. And if something is not clear, Steve says, go ahead and interrupt him. Clarissa will keep an eye on the chat if you want to post questions there. And he will allow about 10 minutes at the end for further questions. So Steve, thank you. And go from there.
All right, thanks for that great introduction. I'm excited to be part of ClinGen, as I'm learning a lot about stuff that I don't know anything about. So here's a slide I'd like to start with. I'm sure everybody here is familiar with this slide, but after the advent of genomics, we were able to assess all the polymorphisms that we see in all of them in the human population and where they're found, basically. And the amazing thing that was found was that if you look at most of the polymorphisms that we see in the human population are shared around the world, right? So 86% of the polymorphisms are in every population. And then only 10% of the populations or of the polymorphisms are restricted to a continent. I'm going to get into that later. And then only 4% are restricted to individual populations, right? So that means that our human population is very young. And so these private polymorphisms are geographically restricted, and they probably represent mutation. And then all these other shared populations are public, and they represent ancient, not so ancient for our species, diversity. And so the question is that I'm interested in is how can we use these differences and similarities to understand human population history? And I'm an HLA person, so it gets even weirder when we start talking about HLA. And so here's HLA. In case you don't know, HLA stands for human leukocyte antigens. All vertebrates have these genes, but in vertebrates that are not humans, they're called the MHC genes, and in humans, they're called the HLA genes. So humans don't have MHC genes, and lampreys don't have HLA genes. And these gene products all present short peptides, which we call antigens, to immune monitoring cells. And this is the basis of the adaptive immune system that made vertebrates so successful. So in humans, the HLA genes are found on the short arm of chromosome 6, and they represent, they're represented by 6 polymorphic loci with these clever names, A, B, and C, DR, DQ, and DP. And this is generally recognized as the most polymorphic region of a human genome. Here's a little cartoon map of the HLA region. There's 2 sections, class 1 loci are A, B, and C. They're about a megabase apart from the class 2 loci, which are DR, DQ, and DP. And there's only one of each class 1 locus, but the class 2 loci are in these different clusters where there's an alpha chain and a series of beta chains. And so there's a DR alpha, DR beta, DQ alpha, DQ beta, DP alpha, DP beta. And when we look at the number of known unique nucleotide sequences for these genes, they're quite high relative to the rest of the genome. So there's almost 8,000 unique HLA-A sequences known, almost 10,000 unique HLA-B sequences known. For DRB1, which is the most polymorphic individual locus for class 2, there's only 3,000 sequences known. But if you look at DQ, where the alpha and the beta subchains, which I'll show you in a second, form a heterodimer, you can have many combinations of 2,000 DQB alleles and 500 DQA alleles to make a wide variety of DQ proteins. So here are the molecules. This is the class 2 molecule and the class 1 molecule. They look very similar because they have the same function. They're both heterodimers. Like I said, the class 2 genes encode an alpha and a beta chain. Class 1 only encodes an alpha chain, but there's a beta 2 microglobulin gene that's found on another chromosome. It's not an HLA gene. But you see the structures are the same. This is the peptide binding groove at the top. I assume you all can see my pointer. I can never tell. There's a pair of alpha helices and a beta sheet that form this peptide binding groove, and the short peptide 8 to 12 residues fits into this pocket, right? So peptide binding groove, and here's the peptide in the cartoon. So here's what these genes actually do. Class 1 HLA genes are expressed on all nucleated cells, so everything except your red blood cells, and they present endogenously derived peptides that are processed in the proteasome to killer T cells, CD8 positive T cell receptors. If it recognizes the complex of the HLA molecule and the bound peptide, if it recognizes that, it knows that the cell is infected with something and it kills the cell, killer T cell. Class 2 HLA is only expressed on some immune monitoring cells, dendrocytes, macrophages, some B cells. CD4 positive T cells look for helper T cells, look for the complex of the class 2 molecule and a peptide that is exogenously derived and processed through the lysosome pathway. And so these are peptides that are found outside of the cell, and that's what these immune monitoring cells are doing. They're looking for weird stuff. If CD4 positive T cell sees this complex of the foreign peptide and the HLA allele, it mounts an immune response. And so this is the brain of the immune system right here. And then I always have to introduce you to our wonderful HLA allele name nomenclature, which everyone loves because it's so easy to navigate. We have the gene name HLA. There's an asterisk, which you don't pronounce. And then we have a series of fields that are separated by colons that tell us about the structure and variation of the gene. So the first field these are historically developed, comes from the days when we were only using antibodies. This is before my time, to determine your HLA type for transplant. So this is called the serologic group or the allele family. So this is the A01 serological group. And then we have the next field is the unique protein peptide sequence for the alleles for an allele in that serologic family. And so this is the second field, the peptide sequence. And then of course the genetic code is redundant. And so we have silent substitutions within a given codon. So we distinguish the peptide sequences with this third field of silent substitution. So this is a silent substitution within a given peptide sequence for a given antibody reactive allele family. And then we have all these regions of the gene that don't code for anything, the UTRs and the introns, and see, these are non-coding variants within this same family of alleles. And so these are non-coding substitutions. And so we have these 4 fields that tell us exactly how this HLA alleles nucleotide sequences is different from another one, basically. And then we have a final thing as an expression variant that tells us if, because of one of these substitutions, it's null, or it's soluble, or it's restricted to the cytoplasm, things like that, which are important for transplantation, but not so much for population genetics. And that the final thing, which is good, is that all these names are assigned by one agency, the IMGT HLA database, which is authorized by the World Health Organization. So there's no ambiguity about what these names are for these alleles, unlike other fields where everyone gets to name their own thing. The number of alleles that we see for HLA increases every year, and that's driven by technology. So when I started in the HLA field 30 years ago, we were doing this PCR-based SSO typing, and now we're doing NGS. And you can see that NGS is really driving discovery of these new allele sequences. But the problem is that based on population genetics, we're expecting that there are millions of alleles at each locus. And so we have a long way to go before we fully understand human HLA diversity. And you probably have seen some of these slides. This is my slide to impress people with how cool HLA region is. It's highly medically relevant. As you might have guessed this, but here's a Manhattan plot looking at chronic hepatitis C virus infection. The HLA genes are found on the short arm of chromosome 6, and here's a short arm of chromosome 6. And when you correct for all these comparisons, the only associations with the HLA region and chronic HCV infection. And that's expected because viruses are pathogens, and the whole point of this immune system is to detect these pathogens, right? Here's systemic sclerosis, which is an autoimmune disease, which is debilitating. And this is, again, associated with the HLA region because your immune system is responding to something in your scleroderma that it shouldn't be. Here's RA, which has long been known to be associated with the HLA region. As you can see, the strongest signal, even there are other signals across the genome, is in the HLA region. I think the p-value for this is in the -40s, just goes off the chart. And even cancers, which they present auto neoantigens, and they want to escape detection by the adaptive immune system, are associated with HLA variation. And then things that were not even thought to be autoimmune, like narcolepsy, which is thought to be neurological, turn out to be autoimmune. And even schizophrenia is looking like it's autoimmune. So that's my plug for how cool the HLA region is. All right, so I want to talk briefly about a couple of diseases. How are we doing on time again? Okay. Nasopharyngeal carcinoma, it's a rare cancer. It's common in China and Taiwan. It's seen much less frequently in Europe. It's associated with Epstein-Barr virus. Almost all NPC tumors have Epstein-Barr virus in them. And then it's associated with eating a particular type of pickled fish that's been pickled with a nitrosamine. And so almost all of us are exposed to Epstein-Barr. I've been to China and I've eaten the pickled fish and it's quite good. But I don't have nasopharyngeal carcinoma. And so that's because it's also associated with HLA-A2 alleles, or O2. This is an old nomenclature. In China, but not in Europe. And AO2 is frequent at about 20% in Southeast Asia and 30% in Europe. So the question for this work we did 20 years ago, oops, that's weird. Oh, one of my slides is, oh no, here it is, sorry. I had them out of order, sorry. The questions was, what's going on in China with NPC? And so here is the result of this work we did. AO201 is seen in China, but it's not associated with NPC. Odd ratio 0.7, not significant. But AO207 is highly associated. You have over twice the odds of developing nasopharyngeal carcinoma if you have AO207. And if you have A1101 or A3101, you're protected against nasopharyngeal carcinoma. So it's clearly an HLA association here. And the interesting thing is that this allele, AO207, differs from O201 only by a single amino acid in the peptide binding groove. And this change, nucleotide, this amino acid change, influences 3 different binding pockets in those alpha helices and beta sheets that presumably affects your ability to present Epstein-Barr virus peptides to the T cell receptor. And in Europe, there's no AO207, which kind of explains why there's no NPC in Europe. So this is a differential distribution of polymorphism that's responsible for regional prevalence of disease, right? That's something that's very interesting, and HLA helps us understand that. And then here's just, these are heat maps. I'm going to be showing several of these. This is the frequency of the AO201 allele in indigenous populations around the world. And so the frequency in here is color-coded, so red is high frequency. The highest frequency is 54% in Northern Europe of AO201, and in the Americas, these are Native American populations. And here you see AO207 is only seen in Southeast Asia. And so this is why NPC is only seen in these populations. And there's also, well, there's other effects, but I'm not going to talk about that. And so here it's differential distribution of a single amino acid polymorphism that's responsible for potentially for regional prevalence of the disease. And if you want to see these maps, you can browse them yourself. You can follow this link. And so the other thing I want to talk about is type 1 diabetes briefly. This is something I've been working on a lot more recently. And we have some known European predisposing alleles and haplotypes. These this list of alleles with a squiggle, a tilde between them, is a haplotype. These are alleles on the same chromosome. And so if you have a copy of DRB10301 and DRB10401 on these haplotypes, you have a almost 17 fold higher odds of developing type 1 diabetes. If you have 2 copies of just the 301 haplotype, your odds are 6-fold. If you have 2 copies of the 401 haplotype, your odds are 5-fold. And if you have just 1 copy of this DRB1 1501 haplotype, your odds are much lower. And so there's a clear effect of specific alleles and allele haplotypes with predisposition and protection for type 1 diabetes in Europe. And Europe has the highest number of children with type 1 diabetes around the world. And so one of the questions is why are these alleles at a high frequency in Europe, and why are they causing type 1 diabetes? And the secondary question is, so here's a DRB1 301, and you can see it's seen all over Western Eurasia and Africa, and but here's DRB1 401, and it's seen outside of Europe, but it's seen in Beringia, Central Asia, and at a high frequency in Northern Europe. And so the intersection of having these 2 alleles in this place is what's causing this high frequency of type 1 diabetes. So one of the projects I'm working on is the Life for a Child project, where we have been looking at type 1 diabetes HLA association in non-European populations. Life for a Child is an international organization that's trying to ensure that no child with diabetes dies from diabetes, basically. And so we want to understand if we can screen with HLA typing for infants and newborns who are at risk for type 1 diabetes outside of Europe. So here's work we published a couple of years ago from Azerbaijan. And you can see in Azerbaijan, if you have a 301, that's predisposing, as well as these other DR4s. And here's a variety of protective alleles, 1502, 1501, which I mentioned. Here's Pakistan. Again, you can see a 301, 1501 is protective. Here, 403 is protective, and that's kind of interesting. And then here is Bangladesh, and again, you see 401 is predisposing, 1502 is protective, and here's Arab population in Sudan, 301 is predisposing, and we have a variety of other predisposing alleles that are not seen in Europe, and these protective alleles. And then in Mali, this was published just this year, 1503 is protective, 1503 is also protective in Sudan, which is interesting. And so when we look at these alleles around the world, you can see that 402 is not seen in Northern Europe, and so it's not predisposing for type 1 diabetes. 405 is seen across kind of equatorial Afro-Eurasia. 1502 is only seen in Southeast Asia, and 1503, which is protective, is only seen in sub-Saharan Africa. So there's different protective alleles around the world. Because these alleles
hey, Steve, can I interrupt for a minute? This is Jennifer Troyer from NHGRI. Just because you went through that really fast, and I'm not as familiar with all the different types and nomenclature. So I think what you were saying, but I want to double check, is that kind of risk alleles are risk alleles everywhere, protective alleles are protective alleles everywhere, but you also see additional different alleles that are seen in some populations, but not others.
That's right. We're seeing this.
But you don't see something that's a risk allele in one population and doesn't seem to associate in others, or same with protection?
That's right.
Okay.
It looks like protective alleles are protective, but they're not evenly distributed around the world, right? So then the question comes where we have population admixture, where you can have someone who has a 1503 and a 401. What is their risk going to be like? And that's something we're going to talk about, but that's one of the things we want to get to with this project down the line. And then a little like 403, which was protective, is seen everywhere except Northern Europe. So one of the things that we want to see is we want to extend this project. Is this allele protective all over the world? That's the question you're just getting to. Or is it only protective in this one population that we've looked at? And we don't have population samples from the rest of the world here to answer this question, but that's something that we'd like to do. Okay, so that's the disease context, right? So these different alleles around the world are causing disease or protecting you from disease. But what we would like to know is what's HLA diversity around the world actually like? And so that's the rest of what I'm going to be talking about. So we have divided the world into these continental regions based on the human diaspora from Africa. And this is most of these are real in the sense that they represent different population groups that seem to be different genetically, but they're kind of also arbitrary for the purpose of organizing data. But it's Sub-Saharan Africa, North Africa, Europe, Southwest Asia, which extends into India, Southeast Asia, Northeast Asia, Oceania, which extends to Madagascar and out to Easter Island, and then Australia. And so we have 144 populations from across the world from this project that we started in 2002. These are the International HLA and Immunogenetics workshops. We just had the 17th one in May this year. So we have them every 3 or 4 years. And we can look at the population size in terms of number of chromosomes, a number of alleles that they have. And of course, if you have a really small population, you're not going to have enough, 2N, you're not going to have very many alleles. But what we see is that once you have a suitably sized population, you have a large number of alleles. For example, look at HLA-B here. These are relatively small population sizes, but they have around 40 HLA-B alleles. And these very large populations, 2,000, 1,000, have maybe 50 HLA alleles. So you're not getting that much more from having a large population size. So it looks like there's a certain number, and you can see that for the HLA-C locus, that number holds up. There's fewer HLA-C alleles, basically, even if you have a large population. So this tells us that there's maybe something that's keeping the allele distribution at your population level at some sort of happy level. We can look at diversity across individual populations. So this big box is telling you the total number of alleles seen in that region. So this is Sub-Saharan Africa. So most populations have between 20 and 40 alleles, but the entire set of population has about 90 alleles. So there's so many alleles that are not shared across populations, and you see that across the world here. Other represents admixed populations, so that we see the same thing, but it's not a real region. And then if we look at the 4 most frequent HLA-B alleles in African populations, you see that they're shared around the world, except when you get to Australia, but they're not at the high frequencies that they are in Sub-Saharan Africa. And then here are frequency maps for those 4 alleles, and you can see they're really generally restricted to African populations with a little bit outside. 5801 is the only one that seems to have spread across Asia from Africa, and it's not seen at the same frequencies that it is in Africa. We see the same thing for HLA-A, except that A shows that A0201 is seen almost all around the world, which suggests that there might be some advantage to having A0201, except for Oceania again, where there's probably population bottleneck or founder effects. And you can see that here, A0201 is very common. This is also why drug companies love to test their drugs against A0201, because it's very high frequency in Europe and seen around the world. We can look at DRB1 and we see a similar thing. There's large numbers of alleles. They're not all shared across all populations within that region. And then the Sub-Saharan African DRB1 alleles, you can see that they kind of tail off as you get further away from Africa. I guess Northeast Asia looks more like Eurasia. And here's more of these heat maps showing kind of the same thing. And then we can look in context at South America, which is kind of the end of the line for human expansion. And you can see that there's 4 high frequency South American alleles, and they're reflected in North America. In South America, they make up about 75% of the allele diversity, and they're not really seen at high frequencies outside of South America. So this seems like it's a population, a founder effect, population bottleneck, and probably with some local selection for new diversity. Some of these alleles, like 411, aren't seen outside of South America. And so here, for example, 411 is only seen in Central America and maybe North America to a certain extent. But 1402 looks like it arose in Siberia and came with the ancestors of modern Native American populations into the Americas. some colleagues of mine, I didn't work on this, but Richard Single and Diogo Meyer did a very interesting study where they looked at 424 Native American populations from North Meso and South America and compared their HLA-A, B, and C and DRB1 allele diversity against genomic microsatellites, which aren't under selection, using the looking population differentiation via FST. And so we can see here that the HLA alleles in these Native American populations are more differentiated from each other than the microsatellites in those same populations are. So that suggests that they're under selection, or some other sort of demographic effect is only affecting the HLA alleles, and so selection seems like our best bet. And so this is just a nice way to show that HLA is different from the rest of the genome, and we can measure that in a variety of different ways. Okay, so what are we doing here? Okay, we have some high frequency alleles that are shared across regions. The degree of sharing differs from locus to locus, and probably each locus has its own kind of idiosyncratic function, or is being affected by selection differently. And then there's low frequency alleles that are specific to certain populations that are probably novel variants that are arising in respect to individual pathogen challenges. And then we see that this differentiation is higher than the genomic background. So HLA is really doing something different. This is not just genetic drift. I'm going to go quickly through haplotypes. These are alleles that are found on the same chromosome. The DRB1, DQB1 haplotype is very small. It's only 50 kb across. And then we have some larger haplotypes we can look at. The AB is the largest haplotype. It spans a megabase because A is so far from HLA-B. And the question is this, these large regions, are they behaving differently than the individual loci? And so here are frequent European DRDQ haplotypes. And you can see this is a 50 kb region with high LD. They're mostly shared with surrounding regions, right? This is the European out to Northeast Asia, Western Eurasia, African region. They have similar haplotype distributions. And then when you get further away from there, you lose those haplotypes. And so there's probably some selection and some recombination as we move out from Africa and Western Eurasia. And then when we look at B and C haplotypes, you can see that there's common European haplotypes, but the frequencies drop off almost immediately when you leave Europe. And that's because these genes are further apart, and there's probably recombination, and the class 1 genes are doing really kind of heavy lifting, so there might also be selection in response to new pathogen challenges. And then when we look at across class 1 and class 2, these are frequent European haplotypes. You see that they're almost not shared anywhere outside of Europe. And so this is probably because of recombination in there's a megabase region between class 1 and class 2. And then we can see, if you look at the common HLA alleles, the individual alleles are shared all around the world, but the haplotypes with those alleles are not. So there's really recombination is driving some of this diversity. And maybe selection for novel allele variants. Oh, and then I'm pretty sure that these haplotypes that we see here that look like European haplotypes, these are actually European haplotypes that were introduced into these populations over the last 500 years. Okay, so that's individual haplotypes. They're more regionally isolated than individual alleles. And that means that they might be more informative for some things because they're being generated at a higher rate. And then this haplotype distribution seems to be inversely proportional to the distance between loci, which suggests recombination. So now we can think about worldwide HLA diversity. I'm going to introduce one more measure, which is how we investigate selection. This is the idea of homozygosity. This is a simple to calculate measure. It's just the sum of the squares of the frequencies. And basically when homozygosity is low, when the allele frequencies are all about the same, basically, there's no difference between allele frequencies. Homozygosity is high when there's a big difference between allele frequencies. So here, the dark red allele goes up in frequency and homozygosity goes up in frequency. So we can have a measure, the normalized deviate of homozygosity, which tells us the number of standard deviations that an observed value deviates from the expected value under neutral evolution, where you have just basically genetic drift and no selection. And that's this FND value. Here's kind of a graphic when you have what we call balancing selection, where the relative ratios of alleles are very even. Natural selection is keeping them all at about the same level. FND is going to be much lower than 0. When we're experiencing genetic drift, neutral evolution, FND is going to be around 0. And when we have directional selection for a specific variant, FND is going to be very high. So you only have a few yellow dots here because we have mostly blue. So here are 3 examples of populations from Asia with 3 very different FND values. So here in Singapore, FND is very close to 0. We have a few high frequency alleles, 1501 and 1502 and 1202 are high frequency, but many low frequency alleles. Here in Java, it's at the end of an island chain, 50% of the population has 1202, which means that basically, on average, each person has 1 copy of 1202. And you can see that there's a strong signal of selection. And we think that this might mean that there was recently or is actively a pathogen, and everybody has to have 1202 in order to survive in this Javanese population. And then here is Mongolia, which instead of being at the end of an island chain, it's in the middle of a continent. And here you have extreme balancing selection. You have all these alleles at very low frequencies. And because there's lots of people moving back and forth through this region, and they're probably carrying lots of pathogens, and so the population has all these alleles in order to protect itself, basically. So we can look at this across the entire globe. This is a study we did where we had 98 population samples. This is just looking at the number of alleles in each population for each locus. And you can see that there's variation in the number of alleles around the world, especially as you go away from Africa, the number of alleles drops for most of these loci. And then we can also look at these FND values for each of these populations across all these loci. And so here is the FND value for the HLA locus. Remember, 0 is balancing selection. 0 is neutrality. High numbers are directional selection, and low numbers are balancing selection. And it looks like there's some slight balancing selection for the HLA locus. And you can see that for the A, B, the CB, DR, and DQ loci. Click too fast. These all seem to be experiencing balancing selection. So natural selection is maintaining large numbers of alleles in these populations, presumably to protect you from large numbers of pathogens. The exception is the DPB1 locus, where there seems to be directional selection at DPB1. And I don't have time to talk about that today, but it's a very interesting question. For DP-alpha-1, we didn't have enough samples to draw a conclusion. But basically, it looks like most of the HLA alleles, most of the HLA loci are under balancing selection for increased diversity. And so the last thing I want to talk about is how we can use all these data to think about population relationships and get to ancestry. And so here's a study we did, again, a long time ago, looking at HLA across Oceania, out of Southeast Asia and into Polynesia. And here we built phylogenetic trees looking at frequencies of DRB1 alleles. And you can see here, we have different regions of Southeast Asia that tend to cluster in the same clades. So here's Australasia. Melanesia is very diverse because it includes Papua New Guinea, and it's been for a long time hypothesized that the native people in the New Guinea Highlands are closely related to Australian Aboriginal populations, and as you can see, that's true here. And then here you can see that this is continental Asia, is closely related to Polynesia, which is also supported by some recent findings of a kind of a rapid movement of these populations from Southeast Asia into Polynesia. We can also, so I should say that this was done just with DRB1 alleles, We can also do this with haplotypes. And so here we have looking at HLA-B, HLA-C haplotypes across a larger number of populations. And I just want to point out here that the distances, the genetic distances here are proportional to these lines. And so we see here that these populations in Africa, Europe, and Southwest Asia are all much closer to each other than these populations. This is the population, indigenous populations of Taiwan. So it's a small island with a large number of populations, and they're as differentiated from each other as sub-Saharan Africans and Europeans are to a certain extent. And Australians are highly differentiated from the rest of these Asian populations because they've been in Australia for 50,000 years. And so the take-home message from here is that our population started here in Africa, and this is Africa, the Middle East, and Europe, and there's been pathogen sharing and probably some degree of genetic sharing for a long time between these regions of the world. But then the rest of the world was colonized more recently, and you can see that as we spread out into the rest of the world, there's new pathogen challenges, and there's decrease in population diversity, which is driving the distance in this tree. So we have these kind of two modes where we have large populations that are experiencing gene flow and probably sharing pathogens, as opposed to small populations that are becoming increasingly isolated and are always seeing new immune challenges. And so that's kind of driving the branches in these trees. Oops. And then the last thing I want to talk about is just the idea of estimating ancestry using using HLA. Whoops, back. And this is just one slide using a program called Structure, where you can give Structure genotype data and ask it to organize the genotype data into a specific number of world groups. And so here I told it to, I gave it genotype data for all of these populations from different regions of the world, and said, organize these into 5 groups. And so we have these 5 groups, which I have called after this analysis: African, Eurasian, East Asian, Australo-Melanesian, and North Asian. And so here you can see, and then I've basically taken all the sub-Saharan African groups and individuals and put them on this part of the slide. So these blue genotypes correspond to these sub-Saharan African groups. And then there's North African and Eurasian set of, I think that's purple. I'm a little bit differently color enabled, shall we say. So I think that's purple haplotypes. And you can see some contribution of these so-called African haplotypes. And you can also see some contribution of these haplotypes coming from Southeast Asian groups. And then you can see moving into the Pacific, these green haplotypes. And then in North American groups, we see these genotypes that are coming from multiple different Australo-Melanesian and East Asian populations, until you get to South America, where it's almost all a subset of these kind of East Asian haplotypes. And then again, this other admixed group looks like it's European, Sub-Saharan African, and Native American. So just to focus on that, this is the Kuwaiti population, and you can see it has a lot of these Eurasian genotypes and a lot of other kind of genotypes from other parts of the world. And then if you look at South America, it's primarily these East Asian haplotypes with a few other genotypes from the rest of the world. All right, so this is my high-level summary. We have high polymorphism for the HLA alleles, regionally differentiated. It's under natural selection, which we think is in response to regional pathogen diversity. And it's shaping these global patterns of allele and haplotype diversity in combination with recombination. And then, so these give us these highly idiosyncratic allele and haplotype distributions for individual populations. But that makes each population really individual, and we can easily differentiate them from each other in many cases. All right, so That's it. I just want to thank my many collaborators. The type 1 diabetes work is what I've been working on most recently. So with Janelle Noble, who was at Corey, and then Graham Ogle and Denira Governor with Life for Child. The NPC work was done a long time ago with my mentor, Henry Ehrlich, and Ray Apple at Roche and Alan Hildesheim at NCI. And I still work with Rich Single and Diogo and Glennis on population diversity and natural selection. Owen Solberg did the work, the heavy lifting of that FND paper. And then, as I said, the International HLA and Immunogenetics workshop is still ongoing. But this work on anthropology was really done with the assistance of Henry, and Alicia Sanchez Mazas and Jose Nunez in Geneva have taken over the anthropology project. They're doing a great job with it. And then I also have to thank Steve Marsh and James Robinson, who run the IMGT HLA database, for giving us one nomenclature instead of having like 15 different nomenclatures, which is a big headache. All right, I'll stop there.
Harry, I think you're muted.
Yeah, I know I'm on mute. I was trying to find the bottom of my thing. Thank you, Steve. That was an excellent presentation. I'm going to open up the floor to a minute to questions, but I'm going to take chair priority or privilege and ask questions that are not related to Ancestry, but related to HLA. So you talked about type 1 diabetes, and from the ClinGen world, we don't have a clinical domain working group yet for type 1 diabetes. And you sort of made a case actually that says, this is the association pretty clearly. Is it that clear? I mean, should we be going down that route?
I think in European populations, it's very clear that there's very specific, very particular, especially DRB1 and class 2 haplotypes that are responsible. I didn't show any of I've also looked at class 1, and it looks like there is some class 1 association, but you have to dissect it in the context of the class 2, the DR to Q.
Interesting. So almost as a subgroup.
Yeah.
So something for us to put on our to-do list then. And then the second question I have relates back to that is, has there been any investigation work into the role of HLA in terms of type 2 diabetes?
There has been, but it's not as clear. It's not a slam dunk the way that type 1 is, and I haven't done that much work with that. I worked on one paper that was it's not out yet because it's not really conclusive. So
okay,
there's other factors, there's other non-genetic factors that work with type 2 diabetes.
Yeah, those diseases, although they are both diabetes, have such different molecular
absolutely
etiologies, right? You wouldn't necessarily expect there to be an HLA effect.
I was just curious. I agree with you, Jonathan, completely. I do think though that perhaps down the line, Steve, once we get the RAD under sort of control and the HLA under control, in collaboration with Jonathan, I think we should have a discussion about type 1 diabetes in particular. Okay, anybody else got any questions? Steve, if you want to turn off your slide, then we can see everyone, and then and if people want to.
There we go.
Oh, we don't need to see your calendar. Thanks.
I'm stopping sharing. Sorry, I got it.
I mean, you're welcome to share it, but I don't think any of us want to see it, or maybe you don't want us to see it. Anybody got anything that they'd like to ask? No. All right. Well, I'm going to ask one last thing, and I'm going to ask you to project ahead. So, as you're aware, I'm sorry, Jennifer, go ahead. I just saw the hand go up. I apologize.
Yeah, no, my hand just went up. So you have.
I thought maybe I missed it, and I was being really rude, and I didn't mean to be.
No, you were not being rude at all. So, I guess the question is, how do you sort of think about using HLA in conjunction with more neutrally evolving markers for population genomics, given that it has potentially different patterns because of selections?
Well, I think we have to dissect them out. I think that if you want something that's completely neutral, You could look at microsatellites or other neutrally evolving, neutral microsatellites, other neutrally evolving markers, and that tells you one thing. But there's
Yeah, I guess my question is how do you think we should be looking at both of them simultaneously when we're thinking about ancestry and populations?
Well, I think that looking at HLA it'll help us understand some of the things that are shaping the events that have resulted in populations managing to survive where they are, right? So, in order to make it in South America, we see that there are new alleles that have never been seen anywhere else, and so those populations experience, especially in South America, some like new pathogens. And there's, in some cases, there's complete turnover of HLA alleles, which you only see in South America. And then we see there for the DP locus, we have a different kind of categorization. You can basically lump all the DP alleles into 4 groups. Some of those populations only have 3 alleles, but they have 3 alleles from all 4 of those groups, right? So it's important to have those 3 HLA alleles in order to make it, basically. And so we can explain maybe why some populations are where they are using HLA, but we can figure out what the relationships are in a neutral fashion with neighboring populations using these other markers. I don't think we can do the same thing with both sets, but I think it's complementary in terms of telling a story and kind of explaining what happened.
Thank you.
Jonathan?
Heidi might have been before
Oh,
me.
I'm sorry, Heidi. You're
Fractionally.
switching on me. My screen keeps flipping. I apologize.
No worries. I was just curious about, there's clear associations. You did a terrific job highlighting so many associations with disease, but do you feel that there's true actionability associated with either screening in advance for HLA types or, antigens, whatever alleles, haplotypes, in any disease context, and or if you have disease already, in a diagnostic setting, knowing the HLA association? You maybe alluded that for the type 1 diabetes scenario, but I'm just curious thinking about HLA haplotypes in general.
Well, yeah, so for type 1 diabetes, I think we can, for a child, my goal with what I'm doing is to be able to come up with a diagnostic. You can, when you have a child born who's in a family at risk, we can say, okay, this child is probably going to develop type 1 diabetes, so we can start intervention. Right, there are things you can do to prevent full-blown type 1 diabetes, right? So we could use a genetic test at birth to start an intervention early. The principal clinical application for type 1 diabetes is bone marrow transplantation, right? And so there we want to really provide a service, or maybe to the larger community in terms of clinical applications. But I think that there are probably other situations where you might think that a disease has a HLA basis, but the alleles are telling you that it doesn't, right? Because we know certain alleles are associated with certain phenotypes. So if you have what looks like one phenotype, but you don't have the alleles for it, that might give you a sign that something else going on, right? That might help you do a clinical kind of subdivision. And then, sorry, you had a third question that I think I forgot.
Well, in that scenario you just laid out, would you treat that patient differently, knowing that they may have an HLA association or not? It's really the actionability of that information, right?
Yeah, I think it's going to, I think it'll depend on the phenotype. And I do know there are examples where having one HLA allele is predisposing for one disease, and having the same allele is protective for a different disease. So it's not a clear cut yes or no. And we have to understand the entire multi-gene context for peptide presentation in order to really to get to that. But here's an example. Chronic beryllium disease is a disease of asbestos miners. And it's because, beryllium is the fourth smallest atom, and it can actually get into the peptide binding groove. And so if you're exposed to beryllium from mining asbestos, your normal native peptides look foreign to your immune system, and you develop autoimmune reaction in your lungs where you've been breathing in beryllium. So you could say, don't be an asbestos miner if you have these HLA alleles. I mean, I think no one should be an asbestos miner personally, but obviously, you see what I'm saying? We can identify risk scenarios for environmental conditions and give guidance on that.
Great, thanks.
Jonathan?
So I know we're over time. If you have time for another question, I'll ask mine. But if you have something else to go.
I'm free for another hour. So
well, hopefully it won't take an hour. So my question kind of is a little bit like Heidi's and sort of follows along, and that is, when we look at associations, there are certainly examples where the odds ratio type enrichment for alleles in people with disease versus those without gives us a predictor, but isn't always the actual disease mechanism itself, right? So whereas you gave an example with one, the beryllium, where there may be a functional a link, right? This allele and this, that it's actually the causal association versus could there be other genes nearby and the HLAs have been selected for, and you've got some other passenger gene that's nearby that's essentially coming along for the ride, but then causing some disease where the HLA looks like it's associated with it. And so how do we differentiate between when the HLA allele itself is the causal responsible party as opposed to some other gene, right, that's in linkage disequilibrium?
We do have an example of that with hereditary hemochromatosis. Originally, it was thought to be an HLA gene, and it turns out it's something in the MHC that's an LD. So their fine mapping is the answer. So we're entering the age, if we are there, where we can do fine mapping across the whole genome. So I think that we're going to see more of that as we move forward.
Yeah. So when we get into kind of how does ClinGen curate, and we think about clinical validity of gene disease associations, an HLA association that may or may not be causative is going to be harder to evaluate than one in which the fine mapping's been done, where you then say, well, the actual causal allele is here, and this is the locus that is the monogenic disease, versus a lot of the HLA-related diseases that might be complex and multifactorial, right, where there is a robust association with HLA, but there are other things that are also involved in that disease manifestation. So
yeah,
it's going to be challenging. Thank you.
That just means many future rounds of grant funding. There's one.
Alice, you want to ask a quick question?
Well, there's one to comment on one thing in the chat. You put it in there. You put it in there, Terry.
Yeah, that was me. I was that was just in reference to Heidi's thing about disease, that HLA is extremely important when it comes. She was asking about, would it make a difference from a clinical perspective? And I was just saying that it does make a big difference, particularly when we think about pharmacogenomics. Alice, you have a quick question? We've got to turn this off in a second.
Yes, thank you. Appreciate it. I'll just share my questions, and Steve, feel free to answer as you will. But my two questions, one was about structural variation. You mentioned kind of an estimate of how much variation you think we're missing. I'm assuming that's sort of like SNVs, and like what impact you think doing high quality sequencing where we can capture all the structural variation is going to impact what we know about HLA. And then my second question is about global diversity. I mean, if we don't have a sense of what all the variation is in HLA genes, what do you think the impact is on disparities in terms of access through matching algorithms and in terms of success of bone marrow transplants, sort of like the clinical applied side of this missing variation for different patient populations?
Well, I think that over the last 30 years, I think we've kind of identified the low fruit for the really common HLA alleles. So, there are, I'm sure there are, I know that there are people out there who have gone through bone marrow typing efforts to go into registries who have a unique HLA type, right? We don't know what the effect of having a unique HLA allele is on an individual. There's probably a reason why there are so many common alleles, because they probably work together, right? Some sort of synergistic way for a given pathogen. So I mean, I think at some point soon, when you're born, you'll have your whole genome sequenced and you'll know your HLA type, right? And then we'll have a huge amount of data to work with and to look at these multi-gene interactions and to get a better sense of what the actual diversity of HLA, functional HLA variance is in the human population, right? So that probably maybe as soon as 20 years away, we'll start to have that information. But until then, there's always going to be this question about this other, two odd million alleles per locus that we don't know anything about. But I get the feeling that for the strong effects with HLA, we've seen most of the strong things, and it's a question of we still don't really understand exactly what how peptide presentation is directly impacting disease. So, for example, with type 1 diabetes, it seems likely that it's a hybrid peptide between two native peptides, like an error that happens in protein processing or peptide processing that's resulting in something that is destroying your islet cells. But we don't exactly know what that is yet. There's a bunch of candidates. So I think that questions like that we can answer, or we can answer for, if we can answer that for type 1 diabetes, those things might be more common for other autoimmune diseases that we don't have a good handle on. I've kind of rambled away from your question. I'm sorry about that. That was the second question. What was the first question again?
It was just about structural variation, sort of the impact of structural variation and how much we're sort of discovering with the Pangenome project and with telomere to telomere sequencing, and how much you think that's going to impact what we know.
Oh, I think that will be very impactful for the genomics. We only have a small number of alt-refs, right? And most of the HLA haplotypes are not represented in those alt-refs. I didn't talk about this, but we have for DRB, we have 3 DRB genes that are on 5 different haplotypes, and those aren't represented in the, most of those aren't represented in the genomic or the alt-refs. So, and that's probably true across the whole MHC, not just for the HLA regions, because it's very gene dense. And so there's probably a lot of structural variants that we would like to be able to elucidate. And there's one example where it seems like there might be a transversion of the class 1 region that is causing complications for recombination, basically, that might be the result, might result in a phenotype. So I mean, I think we need to have more people's full genomes, especially MHC genomes, or chromosome 6 genomes sequenced in order to get a better handle on this. But again, I think that's something that we're going to see over the next, 15 years, I'm hoping. I would like more alt-refs personally for the genomic work.
Great. I think we're going to have to cut it off here. Steve, thank you so much for first off agreeing to co-chair RAD and HLA. I'm self-serving, what can I say? And thank you very much for contributing to the Ancestry and Diversity Forum. If further questions come up, please, you can always contact Clarissa, and Clarissa will get the questions to Steve, and we'll get an answer back to you. Thank you, everyone. See you next month.
All right, bye, everybody.
Bye. Thanks, Steve.
Thank you all. Actually, we will not be having a forum in January because it's the beginning of the year. So we will see you in February.